Guys, welcome you back to my channel Biomixup. Hope you're doing well. So in this today's video, we are going to discuss about the auxin signaling pathway. So the auxin, it is a phytohormone which is present in the plant and it is required for the growth and development of the plant. So in the plant, there are different type of phytohormones which are present, and these phytohormones are being regulated by protein degradation. The signaling is taking place by protein degradation, and the degradation is by polyubiquitination machinery. So first of all, we have to understand how this ubiquitination machinery is required for the protein degradation. Then we will go detail into the signaling pathway. So this ubiquitin protein which is present, it is in the inactive form. So for its activation, it requires energy, and there are different other complexes which are present like E1, E2 and E3. Okay, so this ubiquitin, it undergoes ATP dependent activation by E1. So this E1 is responsible for activating this ubiquitin by utilizing this ATP. Then after its activation, this ubiquitin which is present, it gets transferred into the E2. Okay, now this E2 which is present with this ubiquitin, this complex is transferred into the E3 SCF. Now it forms a complex E2. It makes complex with E3 SCF. And E3 SCF which is present, it has a capacity to target the protein. So the protein which needs to be degraded, it calls that protein towards it. Okay, so there is a binding with this target protein. Now E2 is having the ubiquitin with it. After this target protein attaches with the E3, then this ubiquitin, it is being transferred into the target protein. And when the multiple ubiquitin is present, that means when the polyubiquitin is present, it undergoes 26S proteasome. After that, it gets degraded. So after the polyubiquitination, the target protein gets degraded. Okay, now let us discuss the signaling pathway. So in case of auxin, there is a repressor protein which is known as AUXIAA. Okay, this is a repressor protein. So this needs to be degraded. When auxin is present, this needs to be degraded. Okay, so here we can see that when the auxin is present and there is a repressor over here, that is AUXIAA. It is a repressor which represses the proteins which are required for the growth and development of the plant. So when auxin is present, this AUXIAA, which is a repressor, it needs to be degraded so that in presence of auxin, the growth and development of the plant can take place. So the genes which are required for the growth and development of the plant, they will be synthesized. Okay, so when the auxin is present, it activates this E3 ubiquitin machinery. So this E3 ubiquitin machinery which is present, and like we can see here that E1, this ubiquitin which was present, it is being activated by E1 and it is transferred into the E2. And E2, it forms a complex with E3 ligase complex. So this E3 ligase complex which is present, it is having some other factors like ASK1, CUL1, RBX1 and TIR1. Specifically the TIR1 part which is present, it is having a partial auxin binding site. It is having the auxin binding site. Here you can see that this auxin is binded in this TIR. Near the TIR, this auxin is been binded. Okay. After that, already we have discussed that it is having the property that it will attract the target protein towards it. Here the target protein is the repressor protein which is known as AUXIAA. So this TIR1 protein which was present, it was having a partial auxin binding site and partial site which has the ability that it can bind to the target protein, that is a repressor. Okay, here you can see that after its binding, this E2 which was having the ubiquitin in it, it is targeting this complex or this AUXIAA repressor. So multiple ubiquitin has been provided here. So now it is ready for 26S proteasome by which the AUXIAA is being degraded by 26S proteasome. Here you can see the SCF-TIR ubiquitin E3 ligase auxin receptor complex ubiquitinates the AUXIAA repressor. Okay, so in this way the repressor is being degraded. So let us revise again. So in presence of auxin, the repressor which was repressing the activity of auxin, it needs to be degraded. So for its degradation, there is requirement of ubiquitin molecule. So here the ubiquitin molecule is being activated by E1 by using the ATP, and this ubiquitin is transferred into E2. Now the E2, it forms a complex with E3 ligase complex, and E3 is having CUL1, ASK1, TIR1, RBX1 proteins. Okay. So here in the TIR1, it is having a partial auxin binding site and partial site which can bind the target protein. Here the target protein is the repressor molecule. So now here the repressor molecule is being attached here, and now this E2, it transfers the ubiquitin into this target protein, and due to this there is 26S proteasome and there is degradation of this repressor protein. Okay, now let us see what is taking place in absence of auxin and what is taking place in presence of auxin. So here there is a auxin, due to the auxin response, what happens, the receptor the repressor which was present, now it is being degraded. So here we can see that here what happens in the nucleus, this is taking place, and here there is auxin regulated genes which are present, and here there is a ARF site which is known as auxin response factor, which binds in this promoter region. Okay, it binds in the promoter region and it inhibits the auxin regulated genes. Okay, and this ARF which is present now, it is in the monomer form. It is in the monomer form, and this ARF is being regulated by AUXIAA, which is a repressor. This AUXIAA, it is repressing this ARF because now it is in the monomer form. This ARF which is present, which is known as auxin response factor, it get activated when it is in the dimer form. Okay, now next, in presence of auxin, this is in absence of auxin. This is a condition in absence of auxin. What happens in presence of auxin? In presence of auxin, it activates the ubiquitination machinery, due to which the AUXIAA which was present, it gets degraded. Here you can see that this in presence of this auxin, this SCF, it is the E3. This is a ubiquitination machinery. By this ubiquitination machinery, there is 26S proteasomal degradation, and this repressor is being degraded. After this repressor has been degraded, now this ARF which was present, now it is in the dimer form. 2 ARF is coming and binding in the promoter region, of this DNA, and it is allowing for the auxin regulated gene, and there is expression of the genes which are required for the growth and development of the plant. Here in absence of auxin, there was the repressor which was present, which was repressing this ARF, and due to which there was inhibition of this auxin response factor. This is how the regulation of auxin is taking place in case of the plants for the growth and development of the plant. Thank you for watching this video till the end, and if you like this video, please don't forget to share, like and subscribe. Thank you.